All right, you guys, this is our podcast on neurons. A neuron is the scientific name for a nerve cell. Nerve cells are found in eukaryotic cells. There are three main parts. The first part is called a cell body. The cell body is where the nucleus and all of the organelles are in a neuron. The second part is called a dendrite. The dendrites are like finger-like projections that extend from the cell body, and they are covered by a membrane. And then lastly, our third part of a nerve cell or neuron is an axon. And an axon extends away from the cell body. It's a long membrane-bound projection, and that's going to transmit information away from the cell body in the form of electrical signals. These electrical signals have a fancy name. It is called an action potential. So we will see on how an action potential and a nerve impulse, which is what an action potential is, really works. So axons can be pretty long, but they do end eventually. The end of an axon is called the axon terminal. And so the end of the axon can communicate with a muscle cell, maybe a gland cell, or perhaps another neuron. These axons are covered by a membrane. It's kind of like the cell membrane. And the membrane has a name. It's called the myelin sheath. And ignore ring the bell. We have not learned anything about that yet. The myelin sheath, or the membrane that's around this axon, is going to insulate the axon. And so because of that insulation, it's going to speed up the transmission of these nerve impulses or these action potentials. The myelin sheath is produced by cells called Schwann cells, and these Schwann cells are surrounding the axon. You'll see this all in a diagram in a minute. The myelin sheath, the membrane that's surrounding the axon, isn't continuous. There are gaps or spaces between different sections, and these gaps in the myelin sheath have a name. They are called the nodes of Ranvier. Okay, so here is our diagram of a neuron. So this is what a neuron looks like. You will eventually have a quiz or a test on labeling a neuron. So starting at the top left, we're going to label, and we'll go around. So starting at the top left, those are the finger-like projections that are on the neuron, and that is the dendrites. You can see they have multiple dendrites on the neuron. Going clockwise, we have the cell body. So the cell body is the main part of the cell, and that's where the organelles are. And so you can see that the dendrites come off of the cell body. All right, actually I'm not going to go clockwise. We're going to go down because we just did cell body. The inside of the cell body, because this is a eukaryotic cell, I cannot talk, that is a nucleus. All right, so this purple line that's going all the way to the right, that is the axon. That's where the nerve impulse will travel to. So the nerve impulse actually starts at the dendrite, travels to the cell body, and then down this axon. The axon is covered with a membrane called the myelin sheath, and that is this right here, myelin sheath. Okay, so that's like a cell membrane, a membrane that surrounds the axon that helps speed up the transmission of the impulse or the nerve impulse or the action potential. These myelin sheaths are produced by Schwann cells, so that's the circular thing inside that myelin sheath. Schwann cell. You can see every single section has one of those. And then there are gaps here, 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 and here. Those are the gaps in the myelin sheath, and those are called the nodes of Ranvier. Okay, so the nerve impulse will start at the dendrites, go to the cell body, and go down the axon all the way until the axon ends. The very end of that axon is called the axon terminal. All right, so how do neurons communicate with each other? Because a nerve impulse is going to start on one neuron, and then how does it move throughout the body? It has to be able to communicate with another neuron. So the one thing that's important to know is that first bullet. Neurons do not touch each other. There's a very small space between the neurons. They never touch each other. So the second bullet says there's a small space or gap between the neurons. That space is called the synaptic cleft, and that region or junction is called the synapsis. That space, that synaptic cleft, is where the neurons are going to communicate. There's a signaling activity that's going to happen in the nervous system of eukaryotic cells. And this signaling activity is made up of electrical activity within the neurons, and there is also a chemical flow involved between the neurons. So you have an electrical component and a chemical component. So these synapses do not communicate by touch, but they communicate by releasing chemicals. You might have heard of them. These chemicals are called neurotransmitters, and these chemicals or neurotransmitters get released into that gap, into that synaptic cleft. At the synaptic cleft, or in the synapsis region, the neuron that has the impulse is called the presynaptic neuron, and the neuron that is next, or the receiving neuron or cell, is called the postsynaptic neuron, or the postsynaptic cell. All right, one thing you might not have known is that all cells have an electrical charge inside them, and that electrical charge is different from the electrical charge that is outside the cell. That is called the membrane potential. The membrane potential is the difference between the two charges across the cell membrane. A membrane potential can change if you add and remove chemicals. These chemicals are called ions, and ions are atoms that have a charge. These ions, or chemicals with a charge, positive or negative, can move in and out of the cell. They pass through proteins in the membrane, and they act as ion channels that allow the ions in and out. Whether these ion channels are open or closed, you'll see this in a diagram in a minute, will affect the membrane potential, or that charge that's across the membrane. Just like with batteries that you learned last year, membrane potentials are expressed as voltage. All right, so there's a thing called a resting potential. When the neuron is not firing an impulse, or it's not sending one, it's not receiving one, that neuron is at rest. When it is at rest, the neuron is more negatively charged on the inside, and the outside is more positive. The reason for that is that inside the cell, in the cytoplasm, there is negatively charged proteins, and there is potassium ions in there. Outside the cell, there is a whole bunch of sodium ions. So there is more positive overall outside the cell and more negative inside the cell. That is when the cell is at rest. This is going to change when the impulse goes through. The electrical potential across the membrane when the neuron is at rest is called the resting potential, which I mentioned. And you don't really need to know, but in case you guys are interested, the actual voltage is a negative 70 millivolts at rest. So let's say a cell receives a stimulus, something happens, and so the dendrite is going to receive that stimulus, and the cell body gets stimulated, and then all of a sudden the permeability of that neuron membrane changes suddenly. The stimulus is going to trigger the cell membrane to suddenly become permeable. Permeable means it's going to allow those sodium ions that are outside the cell into the cell. And so the sodium ions that are positive outside the cell are coming into the cell. That's going to change the charge, right? We are at negative 70. We're getting all this positive ions coming into the cell. So the membrane potential is going to reverse, which means inside the cell is going to be more positive than outside the cell, and the outside of the cell is going to be more negative. Because of the reverse in this polarity or this charge, that is when the nerve impulse or the action potential begins. So remember, down the axon, there's segment and segment and segment due to those myelin sheath and the nodes around VA. So the first segment is going to get stimulated, where it's going to change the positive and the negative charges. And then the next segment's going to go, and it's going to travel down that axon from the cell body down the axon to the axon terminal. The action potential is continuing away from the cell body. Soon, the channels that are allowing the sodium ions in are going to close. Okay, so they're only open for a little bit while that impulse is coming through. As soon as that impulse comes through, those sodium ions are going to close, and then the potassium ions, there's potassium ions there as well, those are going to open. And so then potassium is going to leave the cell. And so the whole point of that is we're going to get the cell back to resting potential. So soon after being stimulated, the interior of the neuron is going to become more and more negative and get back to a normal resting potential. That signals the end of the action potential. Little side note here, a neuron cannot generate another action potential or nerve impulse until it has completely returned back to negative 70 millivolts, which is its resting potential. That time, that period that the neuron cannot send another signal is called the refractory period. In order to get the cell or the neuron back to resting potential, it requires energy. So mitochondria is going to be needed to create that energy. And neurons are going to need a continuous supply of this energy called ATP to keep the sodium and the potassium membranes opening and closing and get back to resting. Okay, so this diagram is showing a nerve impulse taking place. You got a zoomed in picture at the top of what's showing what's going on with the channels in the membrane. You can see the impulse is traveling down. It started at the dendrites and then it's traveling down the axon. After it goes to the cell body, it travels down the axon. So you can see the arrow is showing that. And so as the impulse takes place, the negative charges that are normally there on the inside of the cell change to positive. And the positives that are on the outside of the cell change to negative. And so that change of polarity, it's called, or change in the charge, and you can see, travels down from segment to segment, the axon, until it keeps going down to the end of the axon terminal. In the meantime, to get back to normal, because this is already back to normal because the impulse is over here, you can see that there's sodium and potassium kind of swapping spots. All right, a couple side notes. Once the action potential or nerve impulse reaches the end of the axon or the axon terminal, it releases these chemicals called neurotransmitters into that space, into that gap called the synaptic cleft. These neurotransmitters are gonna bind to proteins, and they're gonna open up new channels on the next neuron so that impulse can continue. If enough ion channels are open, the action potential will continue through the next neuron. If not enough ion channels open up, then the nervous signal will be terminated. After the neurotransmitters have opened up the ion channel so that the impulse can continue, they will be cleared out of the synaptic cleft by being reabsorbed by the neuron or just broken down. All right, this diagram looks a little funny. I will label everything so you can see what's going on here, but this is showing you how the nerve impulse goes from one neuron to the other neuron so that the nerve impulse or the action potential can continue through the body. So you have two actual neurons here. I know it's kind of colored. Well, yours isn't in color, I guess, if you're trying to draw this out. I am going to give you diagrams of this eventually. So this right here is the pre-synaptic neuron, and then down here, right here, is the post-synaptic. So the top one is the neuron that has the impulse, as you can see by the arrows. Okay, so here's the axon, and you can see the arrows of the direction of the action potential. So the action potential direction is going on right there, action potential. So here's your first neuron up at the top, okay? And then your other neuron is right there. And so this space right here, okay, the space, that is the synaptic cleft, synaptic cleft. Okay, and then this knob-looking thing, it looks like a bone, that is the axon terminal. Okay, so what really happens is as the impulse reaches the end of the axon terminal, there's these vesicles with neurotransmitters. So all those little red dots in there, those are called neurotransmitters. Those neurotransmitters get out of the axon terminal, they leave, and they get into the cleft, as you can see, the red dots here, and then they connect to these receptor proteins. All right, so this red dot is a neurotransmitter. It's hard for me to fit it in here. Weird looking purple things are proteins on the next membrane. This is open. So if enough of them, you see there's a lot of them here, if enough of them go through, the nerve impulse will continue. If not enough of them open up and those neurotransmitters cannot get enough of them open, then the impulse will stop.